So hello all, we have come with yet another video on amino acids. Thank you for your overwhelming response on the last two videos. I hope you will enjoy this video more. So today we are going to discuss about certain tricks that how can we learn the structure of amino acids, and also on the basis of chemical nature of the side chain, how can we divide certain amino acids. So right on the board today I have mentioned certain amino acids like aliphatic amino acids, OH group containing amino acids. Now we have the sulfur containing amino acids. Now the sulfur containing amino acids are cysteine and methionine. Now what are cysteine and methionine? Cysteine and methionine are sulfur containing amino acids. And one more thing, two cysteines can join together by disulfide bonds to form cysteines. And these disulfide bonds are very important for the stability of the proteins. Alright. Now we have the next amino acids. We have aromatic amino acids with aromatic side chains. So we have WYF, that is your tryptophan, tyrosine and phenylalanine, and due to these aromatic amino acids your proteins can absorb the UV light at certain wavelength and they can give the absorbance value. The absorbance value that we get of the proteins, that is because of your aromatic amino acids. Now we have acidic amino acids. Acidic amino acids I have already told you that have ionizable side chains like Mr. Day, the DE. The DE is having the acidic side chains. All right. Now the basic amino acids. Basic amino acids are HRK, histidine, arginine and lysine. So histidine is what I told you in the previous video to answer which amino acid can act buffer at the physiological pH, that is histidine. And today we'll discuss the titration curve of histidine as well. Okay. So arginine has a guanidino group in its side chain. We will see the guanidino group as well, right? And the only amino acid that has epsilon carbon in its side chain is your lysine. That is also very important. Now amino acid, the most important question, that which one is the amino acid, that is proline. Proline is an amino acid, it is not an amino acid. So it is very important to learn which is amino acid and what are amino acid. It has amino group in its side chain, so that's why it is called amino acid. Now we have amides. Amides are the ones that have CONH2 group in their side chain, RCONH2 group. So RCONH2 group is there in N and Q. So these are forming the amide bond. So now we are going to discuss simultaneously all these amino acids with their side chain in detail, and we are going to also discuss their unique features as well. Alright, now we are going to first discuss about the aliphatic amino acids. So now we're going to discuss the aliphatic amino acid and certain features of amino acid, and I'll also tell you the tricks on how to learn these amino acids, the structures of these amino acids. Okay. Now aliphatic amino acids with first amino acid we have glycine. So glycine has a very simple structure, the simplest structure. It has only just an H atom in its side chain. So it is not forming any chiral compound. It is not a chiral compound. So in the polarimeter, it will not shift the light towards right or left, so it cannot form any D or L configurations. All right, it is optically inactive. So if I ask you, tell me an amino acid that is optically inactive, which does not form any D or L configurations, that is your glycine. It is not having any chiral carbon. Why? Because on these two positions we have hydrogen atoms. We do not have four different groups attached to this carbon, the alpha carbon, the only carbon that is present in this glycine, the main chain, only carbon. So we can say that this is optically inactive amino acid. All right. So these were the most important features of glycine. Next we'll study, when we'll talk about proteins, that it gives sort of flexibility to the proteins because its side chain is very simple. It is just an hydrogen atom. So these kind of things we'll study when we'll talk about the peptides. We'll talk about the secondary structures like alpha structures, beta structures and all of that. Okay. Now we are going to talk about alanine. Alanine is nothing, just in place of hydrogen atom in glycine, you just attach one CH3 group. It will form what? It will form alanine. It will form alanine. It is the first amino acid after glycine that will form an optically active compound. It will, deflect the light of the polarimeter in right or left directions, and on the basis of that we'll get D and L configurations. So it is a chiral carbon. It is a chiral carbon, so it is containing four different groups in its side chains, in the chain, right? It has C double bond O minus here, NH3 here, CH3 here and H here. So there are four different compounds that are attached to the carbon atom. So we can say it is forming a chiral carbon, chiral compound. So it will deflect the light of the polarimeter. It will form D and L configurations. It is optically active. So only optically inactive amino acid is your glycine. Then we have others are optically active. All right. Then we'll talk about the next, these three amino acids. Now these three amino acids are branched chain amino acids. Why do we call them branched chain? Because their side chain is branched. As you can see in valine, the side chain is branched. These three are branch chain amino acids, and there is a very important disease which is associated with the degradation of branch chain amino acid, and there is an enzyme. If there is mutation in that enzyme, it will cause a disease which is known as MSUD, or maple syrup urine disease. Very important question with your exam point of view. Now in the chat section, you will tell me which enzyme is associated with that disease and what are the disease manifestations, what are the symptoms of that disease, MSUD, maple syrup urine disease. Now these branch chain amino acid, first branch chain amino acid we have valine. So you can learn the structure as V. It is forming a V in the side chain. As you can see, right, it is forming a V in the side chain. You can see, right, it is forming a V. A V is being formed, you can see, right? So there is a V in the side chain. So we can say this is valine. Then we'll talk about leucine. Leucine mein two inverted L are being formed. Two inverted L's are being formed, right? You can see, right? Two inverted L's are being formed. So this is the structure of leucine. Then in the isoleucine, we can see the T is being formed. This T is being formed in isoleucine structure. So you can learn it as this. And leucine and isoleucine are basically chain isomers of each other. It is nothing very typical or complex. We can recognize by the name itself, leucine and isoleucine. So they are the chain isomers of each other. All right, you can see that it is having an isopropyl group and it is the chain isomer. Isoleucine is the chain isomer of your leucine. So I have told you already that these are the branch chain amino acids and they are not degraded in liver. They are not degraded in liver. Very, very important. They are always utilized by our muscles. It is utilized. They are always utilized by our muscles, and they are branched chain amino acids. And I've already told you the disease which is associated with their degradation, that is their utilization, that is your MSUD, maple syrup urine disease. All right. Thank you so much. Now we're going to discuss the next category. So our next category is OH group containing amino acids, that is serine and threonine. Now these OH group containing amino acids, as you can see that they have OH in their side chains present. So what can OH group undergo? Like what can we do to these OH groups? They can be phosphorylated or glycosylated by certain enzymes. They can be phosphorylated and glycosylated. In certain enzymes, they are activated or inactivated by the presence of phosphoryl group. That is, they can be activated by phosphorylation or dephosphorylation. Now this activation and inactivation, that is because of phosphorylation on these amino acids. These amino acids can undergo phosphorylation in the active site of the enzymes. All right, they can also be glycosylated by different enzymes like glycosyl transferase. They can also be glycosylated, so they can go phosphorylation, they can go undergo glycosylation. Now one more very, very important thing, that threonine has two chiral carbons. Threonine has two chiral carbons, so it can form diastereomers. It can form what? It can form diastereomers. It has two chiral carbon centers. One is this one. See, four different groups are attached to this carbon, and one is this one. Four different groups are attached to this carbon. So it is forming, it can form diastereomers. There is one more amino acid which I forgot to tell you can form the diastereomers, that is isoleucine. Note that it is very important. The amino acids that can form diastereomers, they are isoleucine and threonine. IT can form what? IT can form your what? IT can form your diastereomers. Very, very important. Just keep this thing in your mind. Okay. Now we're going to talk about the next category. Okay. Now we have the sulfur group containing amino acids. All right. Now in the sulfur group containing amino acids, we have cysteine and we have methionine. All right. Now in the cysteine, we have the SH group containing amino acid, and in this we have sulfur containing. It does not have the SH group, but both are sulfur containing amino acids. Very important thing that two cysteines can actually join together via a disulfide bond and can form cysteine. I have said cysteines can form disulfide bonds and can form cysteine. So we can say that it can form cysteine via the disulfide bonds, and it is very important for the stability of the proteins, as disulfide bonds, confer the stability to the structure of proteins. All right. Now we have methionine. Methionine structure, this is the side chain of methionine. It is sulfur containing amino acid. So we are done with the sulfur containing amino acid. Next we're going to talk about the other category. So our next category is aromatic amino acids. So aromatic amino acids are the ones that have aromatic side chains. All right. So first we have what? We have phenylalanine. Very, very, very easy to learn the structure of phenylalanine. Just to the structure of alanine. This is the structure of alanine. You add the phenyl group. So it will form phenylalanine. All right. So it's very, very easy to learn the structure of phenylalanine. Then to the structure of phenylalanine, you add one OH. Then it will form what? It will form tyrosine. Then to the structure of alanine only, you add this indole ring, then you will form the structure of tryptophan. So WYF structures are very, very clear. Let me just repeat it for you. To the structure of alanine, you add a phenyl group, you will form phenylalanine. Then to the structure of phenylalanine, you add one OH, then it will form your tyrosine. Then through the structure of alanine again, you add this indole group, then we can say that it is a tryptophan. So very important questions from the aromatic amino acids are what? That the indole group containing amino acid. The indole group containing amino acid is your tryptophan. Very important. And tryptophan is that amino acid that actually absorbs the most UV light, the most of UV light at 280 nanometers. Then your tyrosine, then your phenylalanine. Okay, it is very important to know this thing that aromatic amino acid can absorb UV light at 280 nanometers, and the maximum absorbance is shown by your W, then your Y, then F. Let me write it for you. That maximum absorbance at 260 nanometer, 280, sorry, very, very sorry, 280 nanometer, 260 nanometer, which molecule shows the absorbance? Tell me in the chat section. Okay, that 280 nanometer pe proteins show your absorbance. At 260, which biomolecule, which compound shows the maximum absorbance? So W will show the maximum absorbance at 280, then Y, then F. All right. And it has indole group. And one more thing, that serotonin. Have you heard of that compound called serotonin? It is derived from which amino acid? It is derived from your tryptophan. Very, very important thing to remember. So these amino acids are very important to remember. Your F, Y and W. And already I have told you the trick to learn the structures. Now function is that the derivative of serotonin. It is the derivative of serotonin. Serotonin is derived from this amino acid, that is tryptophan. Right, now we're going to talk about the next category. So the next amino acids we have is the acidic side chain amino acids. We have already discussed about them, that they have ionizable side chains. So first one is your aspartic acid, that is recognized by D, designated by D, and your other one is glutamic acid, that is designated by E. So most acidic amino acid is your aspartic acid. And today we are also going to learn about the titration curve of amino acid, as I promised in the previous video. So now we are going to draw the titration curve of amino acid, that is aspartic acid. So just focus on the board. It's very, very important topic, that is the titration curve. All right. So when we draw the titration curve, I've already told you this thing. That on the x-axis, we will add on the OH equivalents, and the OH equivalents we are adding in the form of NaOH. We'll add NaOH, okay, and then we'll add our analyte to that solution. That will be our amino acid ka solution. In this case, we have aspartic acid, all right. Then we have pH on the y-axis. So what happens is, as we keep increasing the pH keeps increasing, right? Then what happened? The main chain COH group will start losing its hydrogen ion into the solution. That means it will actually ionize itself. It will ionize itself. It will donate the hydrogen ions to the solution. So what will happen? This will donate the hydrogen ions to the solution. It will remain like this. Then again there will be a peak. Then again what will happen? What will happen? Then the side chain COH group, the side chain COH group will start losing its hydrogen ion. In the glycine titration curve, what we saw, that there were just two peaks, right? There was one of COOH main chain, then the COOH NH, then the main chain NH3. So these two groups were being ionized, the main chain NH3 and the main chain COOH. But in this one, as we also have the side chain COH, so it will also ionize and it will also give one this kind of thing, right? So we have first main chain pKa, then we have pKr. It is at 2.4. It has what? It has what? It has pKr, that is your 3.6. Then we will come off, then the peak will come off 9.6, that is your pKb, that is your for NH3 group. All right, are we clear with that? Clear? All right. So this is what will happen in the structure of, for the structure of aspartic acid. So we have first 2.4, then 3.6, then 9.6. Now the question arises, these both are COH group, then why don't they ionize at the same pH? Why don't they ionize at 2.4 only? Why? Because there is an additional electron donating group present here. This electron donating group will actually donates its electron to the COH group, and what will happen then? What will happen? Then this COH group will donate the electron, then that electron, that donated electron, that negative charge will not allow the H plus to be ionized from the structure. So this will remain as it is only, and after a certain point of time, then only it will actually ionize and give its H plus to the solution. So this is the titration curve of aspartic acid. So pKa lies in 2.4 range, your pKr lies in 3.6 and pKb lies in 9.6. Now we are going to calculate the pi of the aspartic acid. Very important trick to learn how to calculate the pi of the aspartic acid in the glycine structure. How did we calculate the pi? We added the two pKa values, the pKa A and pKa B, and divided by 2. Then we calculated the pi of our glycine. All right. Now we want to calculate the pi of our aspartic acid. So to calculate the pi of aspartic acid, we have to take two pKa's that are very closely present. Very closely present. Matlab the difference between them is not that significant. For example, the difference between 2.4 and 3.6 is lesser than the difference between 9.6 and 3.6. So we will take these two values and we'll calculate the pi value of aspartic acid. All right. So if we want to calculate the pi value is that pH at which Zwitter ion will form. There will be a point when there will be a Zwitter ion formation in the aspartic acid. So what will be happening? For the pi value calculation, we'll add 2.4 plus 3.6 by 2. So what will it give? 6 by 2, and it will form 3. So the pi of aspartic acid will come at which pH? It will come at pH number 3. So we are done with the aspartic acid and the glutamic acid, and we have also done the titration curve of our aspartic acid. Now we will move on to the other category, that is the basic amino acids. So the next category is our basic amino acids, and these also have the ionizable groups in their side chain, as we have already discussed. All right. So for the basic amino acids, we have HRK, that is histidine, arginine and lysine. Histidine has this group in its structure. This is known as imidazole group. This is known as what? This is known as imidazole group. Tryptophan has indole group. Histidine has imidazole group. Very important. Don't confuse that tryptophan has indole group in its side chain and histidine has imidazole group in its side chain. So if we add imidazole group to the alanine, then we will get what? We will get the amino acid, that is histidine, which acts as buffer at the physiological pH. Physiological pH पे ये buffer की तरह act कर सकता है. Histidine, very important question. It is very important question. Then we have arginine, that is R. Arginine has the guanidino group in its structure. guanidino group in its structure. So this is called arginine. Arginine has what? Arginine has guanidino group in its structure. This is what you have to remember, that which amino acid has guanidino group in its structure, that is arginine. Then we have lysine. Lysine is that amino acid that has epsilon carbon in its structure. This lysine has what? The lysine has epsilon carbon in its structure. So what are the features of these amino acids? That the side chain of histidine is known as what? Side chain of histidine is known as imidazole ring, imidazole group. It is guanidino ring, and it has epsilon carbon in its structure. So these are the basic amino acids whose the side chains of these amino acids can be ionized. And I have promised you yesterday that I'll also draw one titration curve for the basic amino acids. So now we're going to draw the titration curve of histidine. Okay. So let's draw the titration curve of histidine. We'll add the OH equivalents and we'll take the pH values from here. So what will happen? So first of all, on increasing the OH values, the pH will increase. The pH will keep increasing. Then what will happen? The main chain COH group will start ionizing and it will become what? CO minus. So first pKa value comes here, pKa. This COH starts ionizing and gives rise to the CO minus. Then when whole compound, then when this whole becomes CO minus, then what will happen? Then what will happen? The side chain NH group will actually starts giving its H plus ion and it will become ionized. So then side chain, that is pKr, will get ionized for the side chain. It is around pH 6. It is around what pH? It is around pH 6. Then we have the 9.6 pH, that is the pKb. pKb में 9.6 pH पे the main chain NH3 will start getting ionized and it will become what? It will become NH2 and the whole structure will get ionized at this pH 10 to pH 11 तक वो पूरा structure will get ionized. ठीक है? So we have to learn this thing that around pH 6, around which pH? Around pH 6, the R side chain of our amino acid is getting ionized. So we can say that around this pH, it can act as buffer at the physiological pH. It can actually take or donate the H plus ion at that point of time in the physiological pH range. All right. So we have to learn this thing, right? So it is at pH 6, its pKr is there, and at this range, it can actually be act as a buffer at the physiological pH. So this was the titration curve of our amino acid, that is your histidine. Now if we want to calculate the pi, so its pKb is around 9.2. If we consider, it's not important that every time the pKb, that is the main chain NH3, has to be 9.6. In some cases it can be 9.2. Matlab it is in the range of 9.2 to 9.7. It is in that range. Okay, so it is in that range. So in this case, if we take the pKb as 9.2, then we calculate the closely related ones. Then we'll calculate it like this. So we have 9.2 and 6. So 9.2 plus 6 upon 2. It will be around 7.2. It will be around what? It will be around 7 to 7.1 to 7.2. It will come in that range only. So the isoelectric point of this molecule will come around in this range. Alright. So are we clear with that? So that's what. So now we'll discuss about the next category, that is your amino acid. So now we're going to talk about the amino acid. So proline, which is not an amino acid, it is what? It is an amino acid. It is an amino acid. So it provides the cyclic side chain of the structure, actually provides what? It actually provides the rigidity to the structure. Rigidity to the structure of the proteins. What? The cyclic side chain of the proline provides what? Rigidity to the structure. And we'll also talk about that. It is also known as helix breaker. It cannot form alpha helix because its side chain, this cannot side chain, sorry, I'm not saying side chain, the main chain, this amino group cannot form hydrogen bondings. So we can say it is the helix breaker. So it cannot make alpha helices. It is the helix breaker. So we can say it is an amino acid. So what are the features of proline? It is an amino acid. Then it provides rigidity to the protein structure. It provides the rigidity, right? It's cyclic side chain is not that flexible. It is very rigid. Okay. It provides steric hindrance. Okay. So it is providing rigidity to the structure. And the last point, what is the last point? That it is the helix breaker. It cannot form alpha helices. So the amino acids categories are now complete. Now in the next video, now we are done with all the amino acids. We are done with amino acids of biochemistry. Now in the next video, we will start with proteins and peptides. So thank you for your support. I hope you support this video too. And please like, subscribe and share my channel. Thank you so much. Thank you. Bye-bye.